Hello everyone. This week we are going to talk about micropipettes and how we use micropipettes in a microbiology lab. Let us introduce what are micropipettes. Micropipettes are instruments and they look something like this. A micropipette allows us to measure very small quantities of liquid, usually 1 milliliter or even less. We do use micropipettes in a microbio setting because we do want to measure, for example, amounts of broth. Broth is basically a liquid medium, or if we want to go ahead and measure bacterial cultures that are growing in a liquid medium, or we want to measure disinfectants, we gotta use micropipettes if we want to measure any small quantity of fluid. Now, there are multiple micropipettes out there. We call them P, and then I'm gonna explain the numbers in just a minute, but we have multiple micropipettes out there. So we have one that's called P2, there's P20, there's P200. Now, the two that we are going to focus on are the P100 and the P1000. So what does P100 mean? P100, it's a micropipette, and we usually refer to it as the P100. The 100 is actually the maximum amount of liquid that micropipette can measure. P1000, we call it P1000 because 1000 is the maximum amount of fluid that micropipette can measure. The P100 can measure amounts of fluid between 10 microliters and 100 microliters. So the minimum amount of liquid it can measure accurately would be 10 microliters. And the maximum amount of liquid it can measure would be 100 microliters. And so when we come and name it, we go with the maximum amount of liquid it can measure. The P1000, the maximum amount it can measure is 1,000 microliters, and that's why we call it P1000. And the minimum amount it can measure would be 100. Anything in between 100 and 1,000, again, you can set your P1000 so that it would measure anything in between P100 and P100 and 1,000 microliters. Now, when you come and set your micropipettes, you never want to set your micropipettes to anything below the minimum or above the maximum. Otherwise, it is not going to be accurate and you might damage your micropipette. So again, never set your micropipette, and I'm going to show you how you can set it, but never set it below the minimum or above the maximum amounts. Let's talk about the metric system so that we can introduce what's a microliter. The metric system is basically a system of measurement that uses meter for distance, liter for volume, gram for weight. We're going to be talking about volume, so the unit that we use for volume would be liters. Now, this unit, liters, you can make it larger by saying kiloliters, or you can make it smaller by saying milliliters or microliters. To kind of give you an example of how milliliters and microliters, they're actually way smaller compared to a liter. 1 liter is 1,000 milliliters. So you can see how a liter, it's definitely a larger unit compared to milliliters. So 1 liter, it's 1,000 milliliters. Now a milliliter is larger than a microliter. So 1 milliliter is 1,000 microliters. So 1 liter is equal how many microliters? It's actually a million. So there are a million microliters in 1 liter. That kind of shows you how small of a unit a microliter is. It's the millionth of 1 liter. And that's why when we are measuring things in microbiology, because we are dealing with bacteria, which is very small, we usually go ahead and measure things in terms of microliters. Now, to practice metric conversions, try out some of these practice problems right here for you. And by the way, the answers are provided for you on the last slide, but let me just kind of, show you a few examples right here of how you should do it, and then you guys could try the rest later. So let's try this one, for example, right here. If you have 3.5 milliliters, you want to figure out how much is that in terms of microliters. So what you want to do, okay, so you have your 3.5 milliliters, okay, and I'm going to show you how you can, now you can do it in so many different ways. I'm just going to show you one way of how we do it. So you want to go ahead and put your milliliters at the bottom so that it would cancel out, and then put the units that you want to achieve. So I want to have microliters. Now there's a quick trick. The larger unit always gets a 1 next to it. So a milliliter is way larger than a microliter, so you get a 1 next to it. Now 1 milliliter is equal to 1,000 microliters. So you would put 1,000 right here. And then all you would have to do is multiply your 3.5 by 1,000. You're basically going to be moving your decimal point over 3 spots. So at the end, you're going to get 3,500 microliters, and that would be your answer. Now let me show you another one right here. So let's try this one. Let's say you want to go from milliliters to liters. So you have 783 milliliters. Okay, so I'm going to put my milliliters right here. I'm going to go ahead and put my milliliters at the bottom so that it would go ahead and cancel out. Okay, so right here, cancel out, and then I would put the liters up here. Now the larger unit gets to the 1, so a liter is way larger than a milliliter, so I put the 1 right here. Now 1 liter is equal to 1,000 milliliters, so I'm going to go ahead and put 1,000 down here. Okay, so now what am I going to do? Now I'm going to go ahead and divide my 783 by 1,000. Okay, so again, imagine that this is 783.0. Just move your decimal, over on the other side by 3. So the answer is going to be 0.783 liters. Okay. Now you can go ahead and try out the other examples on your own. And as I said, the answers are going to be on the last slide. Let's take a look at the 2 micropipettes that we are going to discuss, the P100 and the P1000. But before I talk about those numbers that you would usually see on a P100 and a P1000, let's go ahead and talk about the parts of a micropipette first. So a micropipette looks something like this. All micropipettes would look something like that. Up here you have a push button, or we usually refer to it as the plunger. And this push button has actually two resistance, meaning two stops. The first stop is usually going to, when you go ahead and press it down, this is what we use so that we can draw up the fluid. Now, if you push down the plunger all the way down to the second stop, we use that when we are trying to eject the liquid that we just drew up. So again, you push down to the first stop so you can draw up the fluid, and then you push down to the second stop so that you can eject the fluid. Right here, there's an ejector button, and I'm going to explain what the ejector button is for. By the way, the push button, you can also rotate it and adjust the volume that way. Or to adjust the volume, you can go ahead and rotate this thumb wheel right here, and you can adjust the volume. Now, how would you know what volume you're adjusting to? There's usually a window right here, and within that window, you can see it displays for you the volume that you are setting it for. So to change that volume, either rotate that push button up here or rotate that thumb wheel down here, and you can go ahead and adjust the volume this way. Now, right here, this is the shaft of the micropipette. Down here, we go ahead and we place a disposable tip. Now, you must place a disposable tip before you go ahead and draw up any fluid. If you forget to place your disposable tip, you are going to damage your micropipette. So it's extremely important that before you drop any fluid, you must place a disposable tip. Now, this disposable tip needs to be ejected before you draw up fluid from another sample so that you won't contaminate your samples. Now, how do we eject that disposable tip? There is an ejector button right here. So this ejector button, once you press down on that ejector button, the disposable tip is going to come off. So whenever you want to, let's say you want to measure some fluid and you have two samples, never go into your two samples with the same tip. Otherwise, as I said, you're going to contaminate your samples. So you have to switch up your tips between, in between samples. And it's pretty easy. You just press that ejector button and it comes off pretty easily. Now, how do you place a tip? All you do is grab on, grab your micropipette, and you're just gonna push the shaft onto a tip, and it usually comes on pretty easily. Okay. Now, how do we use a micropipette? So in order to draw up the volume, okay, indicated by the window. So as I said, like the window right here is going to display how much fluid you want to draw up. Now, how do you read this? I'm going to explain it in a minute, but in order to draw up basically the volume that you want, displayed right here on your window of your micropipette, what you need to do is you need to push that plunger down, and as I said, you push it down to the first stop. Now, while holding it at the first stop, you need to go ahead and basically lower your tip into the liquid that you want to draw, and you slowly release the plunger, Okay, because remember you're pushing it down to the first stop. So you slowly release it and the fluid is going to be drawn up your tip. Now, okay, so now you drew up some fluid. Now you want to eject that fluid out. What you do is you go ahead and you press down that push button all the way to the second stop. When you push it down all the way to the second stop, you go ahead and you eject that liquid that you've just drawn up. There is a virtual lab, and you're going to try out using a micropipette, so it will help you again get a better idea of how a micropipette is used. Now let's talk about how can I read basically, what's displayed right here on my window. Now, as I said, we're gonna discuss the P100 and the P1000. As I said, 100 is the maximum fluid that this micropipette can measure. So a P100, you're going to see two numbers displayed right here on the plunger on the top. So when you look down on the plunger, you're going to see those two numbers. 10 is the minimum amount that you can set this micropipette to measure. 100 is the maximum amount that you can set this micropipette to measure. So this is the minimum, 10 microliters. This is the maximum, 100 microliters. Now, a P1000 is going to look something like this. Up on the plunger, you're going to see those two numbers written, 100 and 1000. So 100 is the minimum amount that you can set this pipette to measure. 1000 is the maximum amount that you can set this micropipette to measure. Again, never set your micropipette to something lower than 100 if you're using the P1000, okay? And don't also set it to anything above 1000, otherwise you're going to damage your micropipette and it's not going to be accurate. Now, if you have, for example, a P20, the minimum for P20 would be 2. So you're going to see like a 2 up here. And for a P20, the maximum is 20. So you're going to see a number 20 written right here. Okay, so as I said, there are multiple micropipettes out there, but we're going to just discuss those two. So let's talk about, okay, so now you understand what that top number means, what that bottom number means. So let's go ahead and figure out how to read the numbers displayed on the window. And we already talked about the tip and how important it is, you guys, again, just to reiterate, make sure that you always change your tip before moving on to a different sample. Okay, now let's talk about how to read that micropipette. So the P100 minimum amount that it can measure is a 10 microliters. By the way, what's a 10 microliters in terms of milliliters? That's 0.01 milliliters, right? The maximum it can measure is 100 microliters, which is in terms of mils, 0.1 mils. Okay, the P1000 can measure 100 microliters to 1,000, or basically 0.1 mil to 1 milliliter. So a P1000, the maximum amount it can measure accurately for you would be 1 mil. Okay, so again, be familiar with how to convert between microliters and milliliters. So go ahead and practice that, okay? So again, try out those conversions yourself and see if you get these these numbers right here. Okay, let's talk about the P100. So the P100, the minimum amount you can set the P100 to measure is 10 microliters. The maximum that you can set your P100 to measure is 100 microliters. Now, the display window is always going to have 3 boxes. Now, if you are using the P100, that first box is going to represent the hundreds. The second is going to represent the tens. The last box is going to represent the ones. So if I go ahead and set a P100 so that my display window is going to show 085, what does that mean? Is that 85 microliters? Is that 8.5 microliters? Is that 850? Okay, so let's see. So if this is the hundreds, this is my tens, and this is my ones, that means that what I'm seeing right here, it's 85 microliters. Okay, now if I want to set it to 100 microliters, then I'm going to set it to 100. So if I set it to 100, that would be 100. Okay, how can I set it to 10? Okay, think about it. So if I want to set it to 10, I'm going to say, I'm going to go ahead and set it so that this box would say 0, 1, 0. So 0, 1, 0, that would be my 10 microliters. Okay, now if I'm using the P1000, minimum that I can set it to is 100, maximum I can set it to is 1,000. So one more time, you're always going to have those 3 boxes. But in this case, your first box is going to represent the thousands. The second will represent the hundreds. The last box will represent the tens. And so if I go ahead and set my P1000 to 073, would that be 73 microliters? No, that would be 730 microliters. Okay, if I set it to 085, would that be 85 microliters like my P100? Nope, it's not going to be 85. It's going to be 850 microliters. Okay, how can I set it to 1,000 microliters? It would be 100. So 100, that would be 1,000 microliters. Okay. How can I set it to 100? 0, 1, 0. That would be 100 microliters. Okay. So again, make sure that you know which micropipette you are using, okay? And even if you set those 2 micropipettes to the same number, like 085, they're both 085. Here, 085 would mean 85 microliters. Here, 085 would mean 850 microliters. And with the P1000, the 1000 is always displayed in a red color. Okay, so again, this would be 85, and this would be 730. You do have to know how to read the P100 and the P1000. So go ahead and kind of, practice. I do give you other examples right here. So there are more examples so that you guys can go ahead and try it out yourself. So if you have a P100 and you set it to 0 to 9, what would that be? that would be 29 microliters. So if you thought 29, then you are correct. Okay, try this one. If it's the P100 and you have 100, what would that be? That would be 100 microliters. 100 microliters, by the way, is the same thing as saying 0.1 mil. What if I say 0.55? That would be 55 microliters. Okay. P1000, remember the thousand, the hundreds, okay, and then the tens. So therefore 0.42 would be 420 microliters. This one right here would be 870 microliters. And this one right here would be 1000 microliters, which is basically the same thing as saying 1 milliliters. For this lab, there's also this interactive micropipette website that I highly recommend that you guys check it out. There are also a few videos that will help you out to see how a micropipette is used in a microbiology setting. And then there's also this other simulation. Go ahead and try it out. And those are all going to help you out see, and try out how a micropipette is usually used. And here are the answers to the questions that I gave you earlier. So go ahead again, practice how you can convert between your metric units. And don't forget to work on the worksheet and try the simulations in the virtual labs on the Connect.